Welcome to Recovering Evangelicals, a podcast for people who were once very comfortable in the Christian faith until the 21st century intruded and made it very hard to keep on believing. I'm your host, Luke Jansen.
And I'm Scott Dyer.
Intelligent design. It's a topic that comes up often in apologetic discussions of faith versus science. We've referred to it often in this podcast and even done a whole episode on the idea. That would be episode number 21, way back in the beginning. And I made it clear that I have let's call it a conflicted relationship with this idea of intelligent design. I mean, the name for this idea is brilliant. What Christian can argue against it without feeling like they're betraying the home team? I mean, if you believe in a God who had anything to do with us being here, it's easy to conflate create with design, and you have to ascribe intelligence to that kind of a creator designer. And so when I'm asked, don't you believe in intelligent design? No matter how you answer the question, you're damned if you do and damned if you don't. My hesitation around embracing intelligent design is simply when I hear the counter-arguments against ID, I have to say that I find them convincing. And so I felt cognitive dissonance for years over this idea. Recently, a friend of the podcast sent us a link to a YouTube video in which Drs. Michael Behe and Stephen Meyer and John Lennox were discussing intelligent design. And the first two of those, Michael Behe and Stephen Meyer, are easily the main proponents of ID. They're the figureheads of the movement. They're both members of the Discovery Institute, whose opening statement for what they do is this: the Institute investigates the life-changing possibilities of a universe brimming with information and intelligent design. The listener who sent us this link wrote in his email, referring to the YouTube video, "This is a long interview, a bit mind-blowing, given the complexity of the conversation by three quite esteemed, highly credentialed academics. In my humble opinion, it's worth investing the time for at least parts of it. If you can find time to screen it, I'd enjoy hearing your comments." Well, I did watch it, and I have to say I had mixed emotions, because those three speakers in the video hit all the selling points for intelligent design, but they didn't address any of the counter-arguments that I frequently hear against intelligent design. So I thought I'd invite one of those speakers on to give me the best responses to those speed bumps. And the natural choice for me was Dr. Michael Behe, because he's a biologist, well, a biochemist actually, among the three of them. So I reached out to Dr. Behe, and he was very kind to make himself available for this. Dr. Behe, welcome to the podcast.
Yeah, welcome.
Thanks very much, Scott and Luke. It's great to be with you. I'm looking forward to this discussion.
Okay. Michael, the setting for that video, I have to say, those were some very impressive digs. You were in a villa in Tuscany overlooking Florence, Italy. Not too shabby at all.
Yeah, I did not pay for it. Yeah, it was kind of a private conference put on by somebody who had the means to do so and who likes to discuss non-standard ideas, stir the pot. And so this time I was invited to it. Apparently it's a yearly affair, but I got to go this time.
Excellent. Yeah, I just want to also mention, really thrilled that you are willing to join us today. I've been a long time following the ID movement for many years. I remember when William Dembski's book came out, The Design Inference, I got that one right away and read it. I can't say I fully understood it, but I understood what I could. And I'm not a scientist, so I'm coming from the lay perspective. But then, of course, Darwin's Black Box, it was a groundbreaking book in the whole movement. And I also read that with enthusiasm. And I went to your lecture. You did a lecture at University of Toronto a number of years ago on the edge of evolution. And I went to that and really enjoyed it. So it's really a thrill for me to have you live here today.
Great. I look forward to explaining things to listeners.
Yeah, great.
Okay, so before we get into the heart of this topic, I'd like to acknowledge your credentials. You're a professor of biochemistry at Lehigh University in Pennsylvania and a senior fellow at the Discovery Institute Center for Science and Culture. And you served as the expert witness in several court cases related to intelligent design, including the famous case Kitzmiller versus Dover Area School District, which hit international news. You've written many books and appeared in many videos and public speaking engagements, all focused on intelligent design, so you are clearly the one to talk to about this topic.
Yeah, I'm getting up there, so I've been in this business for a while, so yeah, credentials accumulate.
Right. Now, I also think it's fair to say in your defense that you're not a young Earth creationist. You fully accept Big Bang cosmology, that life on Earth is billions of years old, and that humans arose through descent from a common ancestor we share with the other primates.
Yeah, that's correct. I think the science on that is pretty good as opposed to the science of Darwinian evolution. So I'm a crank, but only on one topic, not on many topics.
Right. And your connection with intelligent design, your endorsement of it, pertains to certain aspects of biochemistry that you assert can't be fully explained by undirected genetics, at least not yet.
Yes, that's correct. People should realize that back when Darwin proposed his theory of evolution, the cell was pretty much a black box, which is where the title of my first book, Darwin's Black Box, came from. It could do things, but nobody knew how it worked because they didn't have the instruments, didn't have the ability to see down that far into life. So they thought that life was simple, that the cell was a protoplasm, pretty much jelly. And since then, in the subsequent 150 years, science has, of course, discovered that the cell is horrendously complex. It is not jelly. And the point of the book was to say that this is a big challenge to Darwin's theory.
And we will get into that. I just wanted to say one last thing, though. This is not a theological debate. I simply want to look at this idea of intelligent design from a scientific perspective. I think it's fair to say science proceeds, at least in part, it proceeds by scientists making proposals to the larger community, and it's up to the scientific community to ask questions, make counter-arguments, or propose alternative explanations for the same data. And so we have this proposal here, the idea of intelligent design, and I simply want to look at it closely and address counter-arguments that I have heard made against it.
Okay, terrific.
Okay, so we've got a lot of ground to cover. We have corresponded back and forth about the points that we'll be talking about, and I suspect we won't have enough time to get through all of them. But let's begin with the first essential point, some definitions. I don't know if we need to actually define intelligent design, but feel free to do so. But otherwise, this term irreducible complexity, for which you are well known. Why don't you start us off with that?
Okay, yeah. Irreducible complexity, it's a fancy phrase, but it stands for a simple concept. It means that there you have some system or a machine or something that does a function, but it needs multiple parts in order to perform the function. And the example from everyday life that I used in Darwin's Black Box back 25 plus years ago is a mousetrap, a simple mechanical mousetrap that has multiple parts. The one that's pictured in the book, you can buy in any hardware store, got a wooden platform and a spring and a hammer that squashes the mouse and a holding bar that holds the hammer back and a number of different parts. And if you take one of those away, the trap is broken. It's not like it works half as well as it used to. It's broken. And the problem with that, well, I should say that in the cell, science has discovered that, in fact, the cell is run by machines, actual machines made out of molecules. And many of them are similar in that they need multiple parts to work. And in the absence of a certain part, then they fail. And the problem that presents to Darwin's theory is that Darwin says that random changes in selection have to work gradually. You have to start out with something that's doing something, and some change comes along that improves it a little bit, and natural selection then can select it and spreads throughout the population of organisms in the species. And then maybe another change comes along that helps a little bit more, and that spreads and so on. And that's an interesting idea, but there's this class of systems, irreducibly complex ones, where that doesn't seem to work because you need all the parts to do something. And so you can't build it gradually. The only time its function appears is when it's pretty much all put together. And by then, natural selection doesn't have any job to do.
Can you give us an example then of one of these machines?
Well, yeah, in the cell, as I said, perhaps the greatest advancement of modern biology is to discover that the cell, which Darwin and contemporaries thought to be simple, is actually very complex and run by machinery, by literal machines made out of molecules. And I think the visually most intriguing one for most people is something called the bacterial flagellum, which I showed a picture of it in the book. And it's literally an outboard motor that bacteria use to swim. And if you see an illustration of it, it looks like some sort of space age machine. It's got literally a propeller, which is spun around and around and around by a motor, and it's connected to the motor by a drive shaft. And there's another part called the hook region, which acts as a universal joint, allowing the drive shaft to rotate in one plane, and the angle of the rotation is shifted to the propeller. And that's pretty tricky to do. And the drive shaft is attached to a motor which uses a flow of acid from the outside of the cell to the inside to power the turning, just like water flowing over a dam kind of can turn a turbine. And there are other parts like clamps to hold it in the membrane of the cell while it's turning. And just like a mousetrap, if you take away one of these parts, the drive shaft or the propeller or the motor or any of a number of parts, it's not that it's going to work half as well as it used to, but it's broken. It doesn't work at all. So it's real tough to see how a Darwinian mechanism working by what Darwin said it had to do in numerous successive slight modifications could start with something that didn't have a flagellum and build this up gradually. Again, because the function appears only when you've got the entire system in place.
And for the listeners, you referred to a lot of parts. Each of these will be proteins. You mentioned the shaft, you mentioned the flagellum itself, the clamps. You mentioned a number of parts, and each of those are different proteins. Roughly how many are we talking about?
Well, there are at least several dozen. It gets a little tricky because in some species you can have a couple of the proteins kind of stuck together into one bigger protein, or you can substitute a couple. But there are a couple dozen in the physical flagellum itself. And then there's also another bunch of proteins that are needed to control the building of this rather large machine in the cell. But we don't count those in the flagellum itself.
Okay.
So I'm just curious, just as a layperson listening. So do these proteins exist independent of the flagellum? In other words, were these proteins around? So I guess I'm thinking of the mousetrap. So, and I haven't looked at a mousetrap really recently, but I could make the argument that to say, well, mousetraps have a wooden platform. While we have wooden platforms exist without mousetraps. Mousetraps have a spring. Well, springs exist without mousetraps. So in other words, we have all these parts. They come together to make the mousetrap, but they also can be used in many other applications. And so couldn't a Darwinian explanation be possible to say, well, we had all these parts, and somehow through environmental pressures or whatever it is, they start to come together into what eventually becomes this. And I'm sure you've heard this before, so I'm just curious what you would say.
Yeah, that's right. That's a common comeback. Well, maybe these parts were doing other things and they were co-opted, as the phrase is. And I actually discussed this in Darwin's Black Box. Okay. I said, suppose you went in your garage and you were looking around for pieces that might be lying around and that could be used for a mousetrap. And so instead of a little wooden platform, you get a board. Okay, there's a board. Okay. And now you need a spring. I don't know if you've seen the spring in a mousetrap, but it's really rather specific. But suppose you have a spring in an old grandfather clock that you have in the garage. You take that out. Okay. And suppose, you want to use a hammer, a part that crushes the mouse, and say, well, maybe I'll use this crowbar. Well, you start to see the problem. They're not adapted to each other. They're not adapted to working in a mousetrap. And the pieces of mousetrap are not just generic pieces. They're surprisingly specific, and small changes in their shape or their hardness or other stuff, again, causes the mousetrap to fail. One other thing I should point out is that, this analogy does break down, and it breaks down in the sense of making it harder for Darwinian processes, because in the cell, you don't have a human being putting parts together. You don't have somebody saying, well, here's my piece of wood. I'm going to put this spring here and I'm going to tap it down and put in a screw to keep it in place. And I'm going to put this other piece over here. It turns out that the information for how these things stick together in what orientation also has to be built into the pieces itself. So even if these pieces were exactly shaped the same, but they did not have the information for coming together in the correct orientation automatically, as they have to do in the cell, you still have the big problem. Because again, you say, well, okay, let's start with with a mouse or with the platform. Now maybe the spring develops information to have it stick to the wooden platform. Okay, it sticks. That's not going to do anything. Now you get the next piece. Luckily, that sticks, but it's not doing anything either. So in that instance, the problem still exists that you have to adapt these things to their new role in a mousetrap, and you've also got to gradually get them all to stick together. And still, in the meantime, there's no function until it's all put together.
Well, there's no function as a mousetrap, but so for example, you mentioned 5 or 6 parts for this mousetrap, and I do want to get to the flagellum in a minute, but these 5 or 6 parts to the mousetrap, the base and the spring alone could be a door hinge, and then you could take the 2 base plates and a spring, and you've got a clothespin. Mm-hmm. A catch and a holding bar could be the latch for a cupboard door. So those all work. They do things. Each of them do things, and all you're doing is combining 2 parts, and you have a door hinge or a clothespin. And then you could put together one of these clothespins and one of these door latches, and you've got a functional mousetrap. Then you can further finesse it from there.
Yeah. I've heard a similar reasoning from other folks. I was at a debate once at the American Museum of Natural History with a guy named Ken Miller, who is a biologist from Brown University, and he's also a Christian, but he does not like intelligent design as I present it. And kind of to get my goat, he had a mousetrap, and he wore the mousetrap as a tie clip for his tie during this debate. He says, why, heck, I can take out the holding bar, which is the part that keeps the hammer cocked over and stuff, and I can still use this as a mousetrap. And the implication is, well, we could go indirectly towards a function of a mousetrap. And in the debate, I looked out, and there was a lot of folks there who were dressed up in the audience. And I said to the ladies there, I said, okay, ladies, does your date have a tie clip on? And does the tie clip look anything like what Professor Miller wore? And I got laughs like that because I said, well, if he does, probably you're not going to date him a second time. The point is that...
And wouldn't that be selection right there, natural selection? Sexual selection, actually.
Yeah, it certainly would. The point is, if you have a different function, say for a tie clip or a clothespin, it's going to shape the system in a manner that's not going to be compatible with further use as a mousetrap. Natural selection will hone the system to its present use. And just as I talked about, fancying taking a spring from a grandfather clock in your garage and trying to use it for a spring in a mousetrap, you can't, because they're fit for different purposes. So that kind of indirect route, it turns out that people are closing their eyes and they're using their imaginations to guide things in a way that if you look at it skeptically, it would not work.
But maybe let's go back to the flagellum then, because that's ultimately, I want to stick with that example because that's the one that's always been used, and I've heard the counter arguments, and I'd just like to know what the responses are to these counter arguments. You've told us that this flagellum is composed of all kinds of different proteins. I think you said several dozen, and it's like an outboard motor on a boat. But the scientific community are always quick to point out something that I don't hear you mention and address, which is that there's another tool. In addition to that flagellum, the bacteria make another machine using many of the exact same proteins as are going into the flagellum, something that they call the type 3 secretion system. It's a different machine, but it uses many of the very same parts as the flagellum. Those parts fit together much like they do in the flagellum with some important differences. And then this machine, the type 3 secretory system, it also sits in the membrane like a flagellum. And under the right circumstances, it now does something very different. It squirts out proteins like a syringe, like a tube of toothpaste. And this is how the bacteria, they use this machine to infect other cells, their prey. Things like bubonic plague, for example, will do that. So the flagellum is only irreducibly complex if all you want is a flagellum, but you can start with a less complex collection of parts, and you still have a very powerful tool with very incredible evolutionary advantage, this type 3 secretory system.
Yeah, that's fine. Okay, there's a type 3 secretory system that has maybe 10 out of the 24 proteins that make up the bacterial flagellum. And in fact, it pumps out proteins through a tube, a hollow tube, and uses this as a hypodermic to inject poison proteins into its prey. And this is fascinating. And it kind of shows that the bacterial flagellum itself is even more complicated than we had known back when I wrote Darwin's Black Box. That, for listeners, the question is, how does the cell build such a complicated apparatus as the flagellum? It turns out that it's building it from the inside, that in fact, there's a channel and a pump through which the proteins to build the flagellum are pumped up and stuck on the outside of the cell membrane and accumulate and go through a number of layers that we won't talk about. And then eventually the flagellum, and that gets capped off and goes through a very, very complicated dance of constructing itself. Now, the type 3 secretory system uses a protein pump, kind of like the flagellum does. But a protein pump has nothing to do with rotary motion. That is, if you're building something as a pump, you are setting out all sorts of specifications without regard to how it would behave as a rotary motor. So there's no reason to think that this thing should be pre-adapted to be a precursor to a flagellum any more than, say, the ion channels that you study were, or any other function. The two functions have nothing to do with each other. Additionally, in this model, it's the implication is, well, maybe you can build something simpler. That is, something like a pump just pumps out the proteins, and then through some bizarre series of accidents, you also get the ability to rotate a long flagellum and build itself and other things. And so you started with something at least nominally simpler and got to something more complicated. And that would predict that, in fact, the type 3 secretory system was the precursor to the flagellum. But in the past 10, 15 years, a lot of work has been done comparing sequences of these proteins through which scientists who do this thing for a living can try to determine which came first, which came second. It turns out that it seems that the type 3 secretory system is a secondary product of the flagellum. Rather, it seems that the more complicated thing, the flagellum, is older than the less complicated system, the type 3 secretory system. So in that case, that doesn't help explain the bacterial flagellum at all. And what it does suggest is that maybe this complicated system devolved, lost pieces, and then became a functional secondary thing like the type 3 secretory system. And I could envision something like that in a scheme for the intelligent design of life, where you make something first and with the knowledge that over time it'll lose some pieces and pick up a secondary function. But it is not a Darwinian scenario at all to start with something complicated and make something less complicated.
Right. I want to extend the story a little bit more. And in fact, here's how I understand the story. So we have these two machines, the type 3 secretory system and the flagellum, which use many of the same parts. The flagellum has some extra ones, but in both cases, we have machines that pump proteins across the membrane. The type 3 secretory system, one of the things that it'll do is pump toxins, but it can pump through flagellum proteins to generate that whip that it uses. So let's now set those two machines just a little bit to the side of the table, and I want to put a third one on, which is the ATP synthase. It too is a rotary engine, just like the flagellum. You mentioned that. It uses an electrical current passing ions, protons, and potassium and sodium, for example. It also spins at high speeds. And at the end of that rotary engine, not the flagellum, but at the end of this ATP synthase is an enzyme that churns out ATP molecules. So it's a synthetic pump of ATP molecules. It creates ATP molecules. But here's an incredibly important piece of information. The proteins that make up the ATP synthase rotary engine are eerily similar to the proteins that make up the flagellum rotary engine. And again, many of which also make up the type 3 secretory system. And the similarity is found in two incredibly provocative side-by-side comparisons. I want to talk about both of them and get your response. We can look at their 3D shapes of these parts of the ATP synthase versus the parts of the type 3 secretory system, and the organization of the genes for those parts. So let's first talk about the shapes of those engine parts. Those parts are different proteins. They have different amino acid sequences, and obviously when they were discovered, they were given different names because they had different sequences. The scientists thought they were different. But when you look at those proteins from the ATP synthase or from the type 3 secretory system, you look at their three-dimensional shape, they're eerily similar in shape. And so similar, in fact, that you can exchange the parts from the ATP synthase, put them into the type 3 secretory system, or the other way around, and the engines still work. So that's the first amazing similarity. I don't know if you want to comment on that, but then I want to talk about the gene organizations.
You should realize that you're starting with two unexplained things. And ATP synthase, where'd that come from? That's got enormous problems itself. A part that looks like another part. Okay, well, when we build things, oftentimes in our everyday world, we use similar parts, nuts and screws, and even little motors and so on. But you haven't.
If I can interrupt, sorry, but if you mentioned screws. You can exchange screws, but a lot of times the screws won't fit. Even though they have very similar shapes, they'll have a different thread count, thread direction. The screw can be different sizes. So we're talking here about things that not only look similar, but they can be interchanged between those two different machines.
Yeah, that's good. I actually haven't seen that work. So maybe you could show it to me or send me a reference, and I'd be glad to take a look at it. But even if that were the case, and even as you're implying that, maybe this part was just used straight away, or its ancestor was used straight away, there's still, it's like saying, okay, now we've got the correctly shaped spring for the mousetrap. Okay, now we'll put it here. But there are a number of other parts that are not there. And so it's still irreducibly complex. And I suspect that even though we have similar parts and they don't have a sequence similarity, doesn't this suggest that they were made separately rather than the same thing?
Okay, so let's talk about that, the making of these things. Let's talk about the organization of the genes on the chromosome for these proteins that make up either the synthase or this type 3 secretory system. When you look at the genes, they're ordered and arranged on the chromosome, two different clusters of genes, a series of genes that are clustered together that are decoded to make the synthase, the other ones that are decoded to make the flagellum. And you look at the relative positions of the different parts that I just referred to, the genes for those different parts, they show uncanny similarities. The placement, the relative positioning of these things are uncannily similar. Biologists will call this gene synteny. And so where I'm going with this whole argument then is what would be, from one perspective, a very plausible explanation would be that you had a gene duplication, a doubling of a part of a chromosome, which we know that happens all the time in real biology. And so a doubling of the part of the chromosome containing the cluster of genes for the ATP synthase. And others are suggesting that first because it had to come first. It's one of the most fundamental, earliest things that cells need. They have to start making ATP. So a doubling of the section of the chromosome that had the genes for the ATP synthase, and then the duplicate version of the gene cluster could go on and mutate and evolve and eventually produce a very different protein with very similar parts or similarly shaped, the one that's at the heart of the flagellum.
Well, again, you're using your mind to try to guide things. Okay. It's been shown, of course, that the overwhelming fate of duplicated genes is just to degrade. That is, they accumulate deleterious mutations because they're a lot more common than anything else. And that the second most common fate.
Well, let's talk about that one first. You said that they degrade. Why can't we just say that they change and they may lose their ability? The duplicated one, which then begins to mutate, may well lose its ability to do the job that it originally did, but now it's acquiring different functions. And so it's degrading in the sense that if it had to be a mousetrap, it's being a less effective mousetrap, but maybe now it's being a better door hinge.
Okay, well, that is against all of our experience in experimental evolution, in all of the examples that I showed in my most recent book, Darwin Devolves, that when proteins accumulate mutations that what I call degrade, it's not that they're acquiring some different activity. It's just that the activity they had is decreased or eliminated. So, and the reason for that is not anything mysterious. It's just that proteins, as you know, are actually complex systems in themselves, and they have to fold into specific shapes in order to do their jobs. And lots of interactions have to occur within the protein itself, as well as on the surface, to allow it to have its function. And that there are many more ways to mess that up than there are to make something new. So, and again,
maybe that there's more ways to mess it up than to make something new, but that doesn't mean that there can be lots of times where you do make something new. So, for example, I understand it wasn't so long ago that scientists found some bacteria growing outside of some kind of a factory of some kind. They couldn't figure out why the bacteria were growing there, and they found out that these were bugs that used to eat some other carbohydrate of some kind, but they mutated and developed the ability to eat plastic, and they were growing on the backyard dump where they were putting all the plastic. So let's just say, for example, those bugs originally ate starch. Well, now they no longer eat starch very well, and you'd say they've degraded, but they've developed the ability to eat nylon,
right,
plastic.
Right.
That's a great function.
Well, yeah, it is if you've got a supply of it. And but yeah, I know the example that you're thinking of, nylonase. They called it bacteria. It turns out they didn't learn to eat nylon, which is kind of an inert polymer, or pretty inert. But there's a chemical feedstock that is used by nylon synthesizing factories, which kind of spilled out into surrounding waters, and they were able to develop that. And if I remember, it was pretty much just an ester bond that they had to hydrolyze. And so it wasn't nearly as complex as some people make this story seem. I should say that I certainly think that Darwin's theory can do some things. And I do think that you can have mutations that alter activities a little bit around the activity of the parent enzyme, but it's a very limited activity, or very limited set of possibilities.
I just wanted to interject on that note. You mentioned that you do believe Darwinian mechanisms can do some things. So would you count and again, I'm not a scientist, but like speciation, for example. So when organisms over time evolve into other, birds, apparently, we get birds from dinosaurs. Okay. So at that macro kind of level, would you argue that that is more of a Darwinian thing than what we're discussing at the cellular level?
No, in my book from 2019, Darwin Devolves, I said that I think it's most reasonable to say that Darwinian processes can lead to new species in the sense of the lowest biological category. A species like a house sparrow and some other kind of sparrow. Things that you need an expert to tell them apart.
Right.
But I don't think they can. Darwinian processes can explain differences at the level of family, which is the third lowest level of biological classification. I think it can explain species, maybe new genuses too, but not families. And just to give you a feeling, the finches that Darwin collected on the Galapagos Islands, in which a lot of biology texts show, they're all they differ from one another at the species and genus level. They're all finches. They have a smaller, sharper beak, bigger, thicker beak, and things like that. That's the species and genus level. For the family level, you go to things like in the mammalian category, it's like cats versus dogs. I don't think, and there's no evidence to show, that Darwinian processes can create differences at that level of organization.
So where does that leave common ancestry then? What process does explain? Because I believe you do agree with common ancestry, right?
I think common ancestry is reasonable. Yeah.
Okay.
I actually think the information for these things beyond Darwinian evolution had to be added to the system at some point, somehow. And it's interesting because these days, with a lot more DNA sequencing going on, one increasingly common theme that people are discovering is that when they sequence the genomes, the whole genomes of different groups like yeast, there's all sorts of kinds of different yeasts, turns out. This kind over here and that kind over there. And they sequence the genomes to see what the common ancestor of yeast look like. And it's the case that the common ancestor hundreds of millions of years ago had more information in its genome than any of the branches do now, and that these things evolved by losing some of the stuff that they had that their ancestor had. And that I think that goes along well with the idea of you start with a lot of information and you can actually evolve by losing information over time.
Okay.
So let's jump to, I said I wanted to come back to Darwinism. We kind of loosely referred to it a few times in the past couple of minutes between you and Scott. And so I do want to come back to that point because this is another thing that I hear often, that the argument here, the argument for ID is basically that Darwinian mechanisms are not sufficient to explain the changes. And there's always references to Darwinian evolution, which very briefly is just a very gradual evolution, more or less on a base pair level, an individual point mutation, and you just accumulate these over time and eventually come up with something different. And that's more or less the idea, as I understand it, that's captured by Darwinian. But we now know that there's all kinds of other ways to evolve. Many, many mechanisms that involve large genetic changes, not just individual sequential changes that have to accumulate and that don't have to take long, long periods of time. These are large genetic changes that can happen rapidly, sometimes even whole genome changes within one generation. I mentioned gene duplication events. That's another one where you don't just mutate all 1,000 points in the gene to get a duplication. It just swaps. It just copies and pastes a whole segment of a gene or even a whole chromosome, and then you can get reorganization. You get recombination between those two. These are not Darwinian mechanisms. The roles that non-coding RNA that we now know about microRNAs, what they're doing to regulate gene expression. Horizontal gene transfer is another one. Just a moment ago, Mike, you mentioned about E. coli now having more information than their ancestors did. That may be in part through evolution. You referred to devolution, But there's also this horizontal gene transfer, bugs exchanging information. They share genes. And then a huge variety of mechanisms that we call epigenetic changes. The name itself implies these are changes acting on targets other than genes. And so there's so many ways that cells can evolve that don't require individual base changes, which is Darwinian evolution. So why then, if I may, why are you always saying that these are not explained by Darwinian mechanisms? Well, they may not, but they can be explained by other bigger mechanisms.
Okay. Well, you've said several things that I'd like to discuss further that I disagree with. First of all, that's not the definition of Darwinian evolution I'm working from, that only single nucleotide changes, at a time. I think Darwinian evolution is undirected changes. However, the change happens. It's clear that, for listeners, the genomes can be really very big things, like human genome might have 3 billion letters or bases, nucleotides in it. You can change one of them at a time, and that's a mutation. Or, as Luke suggested, you can copy a whole region and put another copy of it right there. Maybe half the genome, maybe the whole genome. I consider that a random mutation. Random mutation, I'd say, is one step, one change. And it might be a single base pair change. It might be a whole duplication, but that's one change. You can think of it as a change in one line of code. Instead of, changing this, you copy a little bit or you copy a whole block, but you're just copying one thing. You're changing one thing at whatever scale at one time. But the point is, it's random, and the point is that you're just changing one thing. And if you're copying things or deleting things, if you're copying things, you're just getting another copy of information you already had. For listeners, let me make an analogy how I see these things. Suppose you wanted to change you started with a book, say Moby Dick, and you wanted to change it into something like Charles Dickens' Great Expectations. Okay. Clearly, you're not going to be able, if I say you can't do that one letter at a time, somebody might come along and say, well, of course you can't do it one letter at a time, but maybe you can make big changes at once. But even if you duplicate a whole page at once, or a chapter at once, or duplicate the whole book, put it tandemly, you still have Moby Dick, and you haven't explained how you're going to get to the new story. So I'm not working from that limited definition of Darwinian. When I say Darwinian, I just mean any random change, but one change at a time. You can't do multiple coordinated changes unless you explain yourself somehow. Right. Just one change, whole genome duplication is fine. One single base pair at a time. That's Darwinian too. And that's what I mean by Darwinian evolution cannot explain these things.
Okay. So, for example, you can have a gene that expresses a protein that doesn't get, or not the protein, but the transcript doesn't get out of the nucleus because it doesn't have the right signal sequence to get it out of the nucleus. So it serves a function in the nucleus to regulate expression of other genes and proteins. But through just simply a copy paste, moving a signal sequence to that gene, now it suddenly gets out of the nucleus, expresses a protein that now does a completely different job. And that's one of the things that I understand happened in the E. coli that Lenski was growing, that basically a promoter was moved. There's a couple examples of this. A promoter was moved so that now a gene which would not have been expressed under anoxic or oxic conditions, now it is, and it does a whole new function. Or another example in a different setting where a gene that was not expressed, a protein that was not expressed because the RNA didn't get out of the nucleus until this signal sequence was just copied and pasted and now it got out. So these are examples of
those things can certainly happen, and I have no problem with it. You have lots of genes, lots of signals and stuff in a cell. And yeah, some gene can accidentally be put over there under the influence of a different promoter, or you could cut it, or you could join it with another gene, or you could accidentally paste things in the wrong orientation. You could do a whole lot of things. And I do agree Darwinian processes, random processes can do things like that, but they don't lead very far at all. The example that you were discussing from Lenski's work is where the gene for a protein involved in allowing the bacteria to metabolize a chemical called citrate, which was normally shut off during the laboratory conditions that they usually used, accidentally got copied and was put next to a promoter that allowed it to be expressed in the presence of oxygen, which is what they used in their laboratory, so that it was turned on. And it turns out that so suddenly these bacteria now could metabolize citrate, which had been available all along, but E. coli can't usually import citrate in the presence of oxygen. So the point is these mutant cells overnight grew up and took over the culture. And it was a big, it was a big advantage for them in their environment. What you usually don't hear about in that story is that prior to that mutation occurring, the E. coli had already lost about a dozen genes through these devolutionary processes that I mentioned earlier. Additionally, after the duplication that allowed it to eat citrate, there were a couple genes that the night before had been working, but now themselves were degraded and destroyed because they interfered with the new environment in which the E. coli would metabolize citrate. And so the point is that for each change, the fastest way to adapt is by breaking genes. That happened before the citrate mutation, and it continued to happen after the citrate mutation.
But you said it yourself that mutation, which, yes, broke a few other genes, but you said yourself that new mutant overtook the culture. It was a highly successful mutant. And that I would not call that a degradative change. I mean, sure, some other pathways that were no longer needed. My understanding of that story, and I've been doing a lot of reading and listening to this story, not all of the mutations are degradations or destructions. Now, some of them would be. It makes sense. So, for example, these bugs are grown in flasks, and the flasks are always being shaken. So these bugs no longer need the mechanisms that they use to move around to look for food. They have cilia and flagella to move around. Well, because they're in the lab, they no longer need those flagella and cilia, and they can lose those. They break down, and other things would break down. You mentioned some mutations that lead to less function as opposed to non-function, but certainly for the purposes that they were originally needed, they aren't needed for that, but they can do other things. So the new functions that they now serve in these mutated bugs is not to do what they used to do, but they take on a new function, these proteins. But the point is, here's an example that you said yourself. It involved moving a promoter into place, and now it could do something that the bugs couldn't do before, and those ones overtook their predecessors.
They certainly did. But what they didn't do is after that, they did not build any new complex pathway, any new complex machinery, such as we see fills the cell, to take advantage of this. As a matter of fact, they continued to degrade genes. In this last paper on this topic, Lenski's lab measured the growth of these things, and they had lost dozens of more genes too. And their death rate, the number of cells that die each generation, had increased from 10% in the ancestor to 20% in the citrate using mutant, up to 50% in the evolved, or the ones that had been adapted to using just citrate over a couple thousand generations. they had not only had the death rate increased, some of the genes that were broken allowed them to take up some of the lipids from the dead cells, the increased number of dead cells in the culture, and use them to in their own as nutrition for themselves. So as I put it in an article I wrote about it, they were reduced to eating their dead, and they looked like they are on the way to extinction. They've lost many, many genes and haven't built much of anything.
Okay, as I said, we're going to be I hope to be talking to them. It's only just in the past week that I've reached out to them, and we will talk to them directly. But we're running out of time, and I do want to cover a couple more points. Maybe we'll move away from the biochemistry and just talk about more philosophical points. The reason I have trouble with intelligent design is because I always have to wonder, what about examples of bad design? If we're going to talk about something happening because of a design, that implies a designer, and we don't use the word God in these discussions, but I think it's obvious that's what we're talking about. We don't have to go there. But this designer is doing these things, and we're ascribing design to that designer. What about examples of bad design? Now, I'm aware of ones on the larger scale, the physiological level, because that was my area of expertise. I worked with airways. And so, very briefly, the fact that we have an airway and the food pipe as one and the same is a bad design. There's no reason that it had to be the same pipe. But in the process of designing it that way, now people choke on a piece of steak. They drown when they go too deep in the water. They can drown in a bathtub. So it's just a bad design. And again, there is no reason for the two to be one and the same. There's evolutionary reasons why they're one and the same, but from a design point of view, there's no reason that we should have the food pipe and the air pipe as one and the same. A few weeks ago, we talked about childbirth, and there's another example where the design is horrible because it turns out that based on the design, some mothers, without modern medicine, some mothers and their babies die horribly because of the way it's all designed. We don't have the time to get into the details. We did that in that episode. What would you have to say then about this idea of intelligent design coming up with designs that either don't make sense or even worse, that they look like bad designs?
Well, I think that's a classic what you'd call argument from ignorance. The argument from ignorance says something like, I don't know why this should be set up this way. Therefore, there's no reason that it's set up this way. And that's not a positive argument. You're not saying, I've exhaustively gone through everything and considered everything and see that there's some alternative that would meet all these goals. You're just saying, I don't know why this should be like that. And that is, people have used that same argument, and it's been shown to be incorrect in some cases. Let me give you an example. You might know that in vertebrates, the eye is supposed to be wired backward. That is, the optic or the nerves coming from the retina don't go backwards into the optic nerve. Rather, they come out towards the front and then circle back into the blind spot, what's called the blind spot, where they all come together and go into the optic nerve through the membrane or through the retina. And that was a classic example of bad design. People would say, that's stupid. Any engineer worth his salt would have had the nerves going backward so that they could not interfere. Of course, it didn't cause any problems because our eyes, vertebrate eyes, are the ones that eagles use to, spot prey from a mile away and stuff. But it also turned out that that in fact, there are other cells which focus light specifically down onto the retinal cells, so that this is actually sharpens the vision in the vertebrate eye, and it also allows the retina to be closer to a blood supply and have the blood supply behind the retina, because retinal cells are extremely active, very high metabolism, and need a lot of oxygen. So the point I'm trying to get across is that saying something is a bad design is not a substantive, or it's not a strong argument, because it just means that the possibility always exists that you just overlooked the engineering reason or other reason why this is in fact put together this way.
Well, as far as the eye goes, though, the changes you referred to, those are changes that are after the fact to improve the vision that was not optimal. And so it's kind of like the Hubble Space Telescope. They had to go up a second time and put basically glasses on the Hubble because the lens wasn't ground out correctly. I just want to finish a couple quick points on this. I think Scott wants to say something, but just I'd like to put these out. The eye, those changes, they're only necessary because of the blind spot, but the blind spot does not need to be there. There's no reason that the blind spot had to be there, but it is there because of evolutionary mechanisms that came up with the eye. And so then after the fact, you have to come up with fixes like the lenses on the Hubble Space Telescope. But the last point I want to get back to, it's not just simply that we look at design and say, okay, it doesn't make sense, therefore it's bad. I'm not making that kind of an argument, which you were referring to. I'm talking about design that is just bad, like the food pipe and the air pipe being one and the same, killing people, and childbirth designed in a way that it kills people horribly. That's a bad design, and there's no reason it had to be that way.
Well, I don't know. I'm not a physiologist or an anatomist like that, but might it have something to do with our ability to speak? And if we had rearrangements in our throat, would we be able to communicate as well as we could? Maybe, as far as I'm concerned, I don't know. But there might be other factors involved that one is not thinking of when you're just saying, this poor lady just died in childbirth, or this person just choked on a piece of steak or something. There might be other reasons, and I think as a rule we should not say, would I have done this differently, or is it evil, or some such, does it cause pain? Rather, we should keep our vision focused on the question, could random changes plus natural selection explain anything? Could it explain a throat at all? Could it explain childbirth at all? Could it explain an eyeball at all? And for reasons I lay out in my books, I don't think it could explain any of those starting points either. So,
Okay.
yeah, so
Just to put a cap on that. I do tend to agree that a good or bad design is a value judgment, right? And it is according to the values of the person who's they deemed it good or bad. I mean, I'm sure we've all had the experience where maybe we have to do something on our car, and we think to ourselves, my goodness, what engineer in his right mind would have put this there when I got to take apart 50 things? Well, the thing is, I'm not the engineer, right? And there very well could be a whole bunch of really good reasons why they did that. I often think it's the good reason is the profit of the car company. But nonetheless, I do tend to agree with that. But not to say that things like childbirth and so on certainly make you wonder. I mean, as you were talking, I started to think on the childbirth. One of the major steps, apparently, in primate evolutionary behavioral development was the whole fact that humans bond, right? They bond with their babies. They bond with the mother of their babies. Could it be that a terrible childbirth caused men to bond more with the mother? Because they experienced this together with her. They had to take care of her, I would think, back in very prehistoric times. So could that have been a benefit? So you could probably shoot a million holes into that too. But the point is that that's not so much a scientific argument. I tend to see that. But here's where for me, if you don't mind, my biggest fundamental issue, I guess, that I like to hear Michael address is the scientific nature of IDs. So let's suppose that you're totally correct, that current random mutation Darwinian things cannot give rise to complex machines and so on. Well, then the question becomes, from a scientific point of view, well, then what does? And this is where I think the critics have a real hard time in that, well, I get the sense that the ID people would say, well, there's some sort of intervention. They may not want to call it divine, but there's been some sort of, somebody has rigged the game. There's some sort of design going on there that we don't understand. But does that put it beyond science then? In other words, is it fair for the critics of ID to say, well, okay, but that's not science. That's something beyond science. It can't be scientifically demonstrated. Therefore, intelligent design, it may be true, but it's not science. So how would you kind of, and I think the Kitzmiller case, the judge was pretty scathing, I believe, in that case, saying that there was just no scientific evidence for any of this. So how do you address that?
Okay, well, let me address it by citing the example that I talked about for a long time in the court case, and which the judge completely ignored because, in his opinion, he simply copied the written document that the plaintiff's attorney gave him, and he showed absolutely no evidence of grasping any of the arguments that were presented in the trial. Again, he simply copied it. We can go into that if you'd like. But here's the thing. You say, is the beginning of how does information or what does happen, how is the design affected? Is that off bounds to science? Say, no, go at it. Do your worst. I think it might be a tough question to address, but that's not unique. This is not the first time in the history of science. And I think a good example that I talk about in the trial is the Big Bang Theory. If you remember, in the 1930s, before the 1930s, most physicists thought that the universe was eternal and unchanging. And then the motion of galaxies away from each other, made it look like we're in the aftermath of a big explosion. And if you roll that back in your mind, then a long time ago, all the matter in the universe was together, and that suggested a beginning to the universe, the Big Bang Theory. When it was first proposed, a lot of physicists hated the Big Bang Theory because it seemed to have theistic implications. I think it does have theistic implications. A lot of people will admit it. But nonetheless, everybody thinks the Big Bang Theory is a scientific theory because it's based on physical evidence, our observations, plus normal logic. If you turn back the clock, you see the matter going back into a small space, as if in a Big Bang. Now, how could a universe explode into existence? Is that off limits to science? Well, no, go at it. People still don't know how that could happen, but they're working on it these days. Haven't come up with an answer yet. I'd say the same thing for intelligent design. It's a strong hypothesis based on the physical evidence, the existence of sophisticated molecular machinery at the very foundation of life, and so on, plus ordinary ways of reasoning. Whenever we see purposeful arrangements in machinery and mouse traps, we always conclude that it was purposely put together. Maybe in the future, somebody will figure out how that could have been done, how the design was affected. Maybe it has theistic implications, but that's what the physical evidence say. Let other people worry about the implications of it. The evidence points to design.
But you put side by side these two theories, these two proposals, one being the Big Bang Theory and one being intelligent design. And in both cases, we can't really see the endpoint yet, but there are people who are thinking, in both cases, people who think, okay, I think that's what's going on. The difference is in for the Big Bang. You have labs around the world doing all kinds of experiments, collecting data, making predictions, which then are either verified or not, and then they go back and try some new experiments based on that. Making predictions, testing the hypotheses, and doing things, coming up with data, number crunching. On the ID side, I'm not aware that there are experiments being done, predictions being made, hypotheses being tested, data collected, papers being published. I'm not seeing that.
Mm-hmm. Well, a couple things. First of all, very few people are working on the Big Bang in the sense of seeing how it originated, as you say. There's not that many experiments being done saying, how does universe explode from nothing? There aren't many, well, there are some theories, but certainly the very great bulk of physics is not working on that. What they're working on is the aftermath. Okay, given a Big Bang, and now things are spreading out, how will the universe develop? You could say, okay, well, looks like this and this and this in biology require design. Okay, given that that's design, how will it play out from there? And I think that's what a lot of laboratories are doing. That's what they are doing. They essentially work from a design perspective, but they don't call it that. They go on and say, well, let's look at this system. This system works this way, but it has to be controlled. So let's look for a control mechanism. And they go and search for find a control mechanism and kind of spell out the workings of engineered type systems. You know what biologists are not doing? Biologists are not working on how the flagellum might have developed randomly by random changes and natural selection, or how ATP synthase could have developed by random changes and natural selection. The closest anybody's come to it is, again, I say will be is the work of Richard Lenzi's lab. And when you do experiments like that, you find that evolution does a whole lot less than people's imaginations have thought for the past 100 years or so.
We didn't have time to get into it at the time, and I really wish we could have. We just don't have time. But I have come across the papers where they're looking at and making predictions and doing experiments to show that you have this sequential evolution between the ATP synthase, the type 3 secretory system, and the flagellum, and for that matter, structural proteins that make up the cytoskeleton. So there is that work.
So when you say guided, I'm curious, I guess you're certainly open to discovering some sort of unknown but scientific, empirically verifiable mechanism that's guiding the process. Yeah. Or the alternative would be, I guess, there's some sort of outside influence, namely God or whatever, guiding only at certain points along the, not the whole. So I'm curious, just the whole guiding thing. What's
up with that? Short answer is I don't know how this is guided. I'm certainly open to discovering an empirically detectable mechanism for guiding, but I don't think there is one. Okay. Simply because a mechanism implies something relatively simple, relatively has a few parts, and if you bump up against there, you'll get knocked back. And I think to explain all the information in cells, you need to specifically add, that information. When an engineer makes a new motor or an air conditioner or something, they don't have some mechanism that designs it. They have to plan out all the parts and how they fit together and stuff.
Right. Okay.
I don't necessarily think there were spurts of guidance. For all I know, everything might be guided all the time.
Okay.
But the only question I'm interested in or focused on is what is empirically detectable. You can guide things where you can't see what's, looks pretty normal to you.
Right.
But it's only in the instances where things don't go as you expected that you realize something extra is going on. But it might have been there all the time, as far as I know.
Right. Okay.
Okay, we are long on time. I'd like to make a statement, and I'll give both of you opportunities to make a statement. But here's the last one that I'd like to make. I just want to get your thoughts on this. obviously I do get animated about this, and the reason I get animated is because I do believe that there is a creator. And you'd think that a creator God would be intelligent. So I want to believe in it. And yet I see these examples of what I do think are malevolent or bad, not just bad as in clunky, but even malicious designs. We've already talked about that. Here's what I wanted to say, though. I think this concept of intelligent design, it forces people into a difficult decision that I don't think they have to make. Mike, you were the expert witness at the Kitzmiller versus Dover case decades ago, and there was a lot of consideration of whether ID is a religion rather than science. We've kind of talked about that already. And another expert witness at the trial, Dr. Ken Miller, he said that his concern was that ID forces people to choose between religion and science. And then we come to today, 20 years later, the YouTube video that I referred to at the beginning of the interview where you were in Tuscany talking with Stephen Meyer and John Lennox about intelligent design. And the host of that conversation started the whole discussion off with these 6 words. He said, "Who's dead, God or Charles Darwin?" And that's the problem for me. I feel like this forces people to choose between religion or science, and it is a zero-sum game. If you choose the one, then you have to reject the other.
Well, okay, let me just briefly comment that I do not view it as choosing between science and religion. I am a Roman Catholic, and I was taught Darwinian evolution in parochial schools. I never had any theological difficulty with it. Could be God's way of making the world. I have scientific difficulties with it. So I don't want to reject science, and therefore I do not automatically accept Darwinian evolution because the evidence seems to be strongly against it.
In the scientific community, are there others not coming from any, you said yourself, you have a Catholic background, but are there others coming from, are there even atheists who are starting to question some of the things that you've raised?
There are certainly some atheists who are very skeptical of Darwinian evolution and who will say, they don't believe in God, but we sure don't have a clue as to how this life came about. One prominent one is a guy named Thomas Nagel, who's a philosopher at NYU, and he even wrote a book essentially dissing Darwinian evolution, and he got a lot of criticism for that from the Darwinists. But yeah, there are some, although certainly not as many as Christians.
Yeah.
And my rebuttal to what you said about how there are scientists today who have not got a clue how this all happened, I get that totally. An analogy that I've used in the past would be humans flying. There was a time, it was only a couple hundred years ago, where people said there's no way, absolutely no way humans will fly. It's impossible. It doesn't happen. It will never happen. But we eventually learned a few things about aerodynamics, and now we're flying all the time. So we just have to wait and see what we're going to learn in the future. Scott, did you have anything else you wanted to cover before? I think we really need to get close to a close.
No, I know we do need to kind of wrap up, but I guess, yeah, I mean, this leads into a whole other discussion. Maybe we should have another time, but kind of a philosophical discussion of science itself, right? What is the nature of science? And is there an edge to science? You wrote a book called The Edge of Evolution. Is there an edge to science? Let's suppose we do pretty much unequivocally prove the Big Bang, although now apparently the James Webb Telescope is throwing some curveballs on that one. But suppose we do. Could we get behind that? There's a famous quote attributed to both Aristotle and Augustine. What was God doing before he created the universe? And the answer being creating hell for people who will ask questions like that. So it could be that we get to the Big Bang, or that we get to the at a certain point in the development of evolution where we say, you know what, we're at the edge of science here. We can't really know beyond this. I don't know. I'm not saying we're there or we're even close to it. The multiverse comes to mind as well, right? Because, there's a lot of debate. Is that a scientific theory? Well, not really, because you can't empirically test it, right? So one thing that I would be curious about is, what are the experimental designs that would lead us to prove scientifically design? What experiments could falsify, I guess, to use the falsification idea? What experiments would falsify the design idea?
Well, that's a great question. And essentially, it's the flip side of the design argument. I say that intelligent design is required to make complex interactive systems. So if somebody like, say, Richard Lunsky went in and grew bacteria and kept their hands off, and over time they saw that, some new propulsion system, maybe not the flagellum, maybe something else, or maybe some new metabolic machinery was made, then my claims would be falsified.
Right. Okay.
But we are out of time. Mike, I want to give you the chance to have the last word. Anything, if maybe we didn't talk about, or how you'd like to sum up?
I'll make one comment, and that is that question of what is design and how do we recognize it? We actually didn't get to talk about that. And briefly, we recognize design. We can't read minds. So we have to reach a conclusion that something was designed or that some mind has acted by looking at physical stuff, by looking at physical arrangements or whatever. And we recognize design by a purposeful arrangement of parts. When we see different parts that are put in relationship to each other so that they can do something, like in a mousetrap, that's how we recognize design. In the past 100 years, modern biology has discovered deeper and deeper and deeper levels of purposeful arrangements. And that is what the modern idea of intelligent design is based on. It's not primarily a religious conclusion or a philosophical conclusion. It arises simply from our ability to recognize the work of a mind in a purposeful arrangement.
Okay.
Thank you.
With that, Michael, I want to thank you very much for coming on to our show.
Yes.
I'm not sure that either of us have convinced, between the three of us, we still have many differences, but we've given the listeners a lot of food for thought, and we'll just leave it with them. I hope you are up to coming onto the show again if we have some follow-ups.
Yeah.
Okay. Thank you so much, Mike. I really do appreciate it.
Yeah. It's great to talk to you. Okay.
Have a good weekend.
Okay.
Bye for now.
Thanks.
Bye-bye.
Bye-bye.
You've been listening to Recovering Evangelicals with your hosts, Luke Jansen and Scott Dyer. Send your comments and suggestions for new episodes to lukejjansen@gmail.com. Join our private discussion group on Facebook by searching for the Recovering Evangelicals podcast group. Look for the logo that you always see at your favorite podcast catcher and ask to be let in. Subscribe to lukejjansen.wordpress.com if you want to get advanced releases and group posts, or browse through our archive for a full listing of all our previous episodes. And please help the podcast by sharing with a friend and leaving a review at your favorite podcast catcher.